So our first example of uh, a normal accumulation of intracellular accumulation is a fatty change, also called steatosis, steatosis. I've heard it pronounced several ways, but steatosis is referred to as fatty change. And fatty change can happen in liver, heart, kidney, and muscle. Well, these are the four most common places that it can happen, and the most common is inside the liver. And why? Because the liver is the organ that is mostly responsible for fat metabolism. Fat metabolism. So the liver is where most of the fat um, is metabolized in our body. And steatosis can be caused by toxins, protein malnutrition, diabetes mellitus, and obesity and anorexia. And in industrialized nations, the toxin alcohol and um, diabetes mellitus combined with obesity is the most common um, circumstances by which we see a fatty liver or steatosis. So I've drawn here, or I've added some molecules, and we're going to discuss some concepts about the liver here. Let me just scroll down here a little bit. Okay, so if I were to draw, let's just say all these concepts are inside of a liver here. This green outline is supposed to be a liver here. So free fatty acids, free fatty acids are introduced into the liver, um, either from catabolism or breakdown of some sort, from a fat cell that's somewhere in the body, or they are brought to when you they're brought to the liver through digestion. So when you eat a big meal that has some fatty acids in there, they're brought to the liver um, after absorption through the gut. So they come up in here. So from the gut, from body, somehow, whether it's a breakdown of fat. Let's just add in here adipose tissue. That's usually where it comes from. It's from the body, but either way, it's from the adipose tissue inside your body, or the fat tissue, where the fat's stored in your body, or from the gut. The free fatty acids come inside the liver, and they are converted to. Let me get the right color here. They're converted to triglycerides, and triglycerides then are converted. They undergo catabolism, which is converting, a molecule like this into a molecule like this. That's catabolism. This is the process of catabolism, is converting a molecule with a lot of different parts to smaller parts. So that's what catabolism is. So the free fatty acids are converted to triglycerides inside your liver, and the triglycerides then are converted to cholesterol. And cholesterol is very important to your body. Um, you know, the cell right here, we've talked about, we've drawn a lot of pictures of cells, but the cell is composed of a membrane that has two layers, and inside this cell membrane is collect little, globules or little clumps of cholesterol, and that helps maintain kind of a fluidity of the membrane so that the membrane isn't stiff. It kind of helps the membrane be able to adapt and can go in, it can go out and not break in half. So cholesterol kind of helps maintain the fluidity or the malleability of the membrane. Phospholipids are also obviously a big part of the membrane. If I were to zoom in here, you would see the outside and the inside, and then you'd have these little phospholipid tails here. And phospholipids are these little tails here. So phospholipids are a big part of your membrane in every cell of your body. Or triglycerides can be broken down or oxidized to ketone bodies. So you have, I'm going to draw a triglyceride here. So here's my attempt to draw a triglyceride. So every triglyceride has an ester. This bond right here, where a carbon is double bonded to an oxygen, then singly bonded to another oxygen, that's called an ester. So there's 3 esters, and then they got these long tails, these long fatty acid tails here. And I'm sure you've heard of unsaturated and saturated fats. And the unsaturated fats have some kind of double bond, an alkene or a double bond inside the tail here. And a saturated fat is the one that just has hydrogens, but there's no double bond. They're just hydrogens all the way down. And an unsaturated fat has double bonds inside of it. And in our video about free radicals, these reactive oxygen species come in here and they attack these double bonds, and you can review that video to kind of see what happens. Also, if you want more information or learn more about unsaturated and saturated fatty acids, then you can just Google that. But so these fatty acids, these triglycerides here, they're broken down into oxidized to ketone bodies. And the 3 most common types of ketone bodies are acetone. Acetone, that's acetone. This is acetone. Or you have acetoacetic acid, which is this functional group is a carboxylic acid, and this is a ketone. And then the beta hydroxybutyric acid, instead of a ketone, I believe this is an OH, but I'm not quite certain. You can Google that to get the exact chemical structure. But the idea is this oxidized to ketone bodies this is what acetone is a byproduct. So this one goes out in waste. Body can't use that. But this acetoacetic acid and this beta hydroxybutyric acid, these 2 structures here, this video is getting kind of jumbled, getting kind of a lot of parts in here, but this acetoacetic acid and this beta hydroxybutyric acid, those are used in the brain and in the heart as energy. So when you're in starvation mode, either you're fasting or you haven't eaten in a long time, or you're out in the wilderness, Man versus Wild, or these reality TV shows where people are out in the wilderness trying to survive, their body is undergoing this process a lot because they don't have a lot of protein, they don't have any carbohydrates, so they're metabolizing all their fat. They're turning all their fat into usable energy, and this acetoacetic acid and this beta hydroxybutyric acid are the compounds by which your brain and other parts of your body use the energy. And finally, these triglycerides are converted into lipoproteins. I don't know if that red shows up very good, but the triglycerides then are converted to lipoproteins with the addition of apoproteins. So the apoproteins combined with the triglyceride equals lipoproteins. And I'm sure you've heard of, when you go get your cholesterol check, you hear the LDL or HDL. These types of molecules, these are lipoproteins. These are a class of lipoproteins. And there's that's not a very good VV, LDL, there's chylomicrons. There's different types of lipoproteins, and these lipoproteins can then be excreted into the blood for transportation. So this is the way that triglycerides get transported to other tissues inside your body, is through these lipoproteins. So the same idea with that manufacturing plant idea is that if you have Department 1, Department 2, Department 3, in the last video, if there is problems in any of these pathways, then you get intracellular accumulations. You get these, let me scroll up here. You get this fatty change, or this steatosis, or steatosis, however you would like to say it. You get this process of where you start getting this fatty liver, or the fatty kidney, or the fatty muscle, or the fatty, organ, because you get these triglycerides and these little fat components building up in your cell. So some of the examples is, this is oxidation. So oxidized to ketone bodies, these triglycerides can be oxidized to ketone bodies. Needed to make more space in here. Well, let's do this one first. The apoproteins, if you have carbon tetrachloride, which is a toxin. So if you ingest some carbon tetrachloride, or you have protein malnutrition, then you're going to decrease these apoproteins. So if you don't have these apoproteins, then these triglycerides are not going to be converted to lipoproteins. So you're going to have accumulation of triglycerides inside the liver, which will cause fatty liver, or steatosis. So if you ingest carbon tetrachloride, or you have protein malnutrition, or you're not consuming enough protein, then these triglycerides are going to be consuming inside your liver. And then let's talk about these oxidized to ketone bodies. So you have anoxia, which is without. This means without. Oxia is oxygen. So if you are without oxygen, you're not going to oxidize these ketone bodies, because in order to oxidize something, usually you need O2 within the body. In chemistry, there's tons of ways to oxidize other things, but within the body, to oxidize something, you usually need oxygen. So if you don't have oxygen, you're not going to oxidize these ketone bodies. You're not going to oxidize these triglycerides to ketone bodies, and then you're going to have a buildup of triglycerides. Another way for a problem in this oxidized of ketone bodies is alcohol. People that drink too much alcohol, it inhibits mitochondria. So we talked about mitochondria. This is a picture of a mitochondria. Inside the cell, you have all these mitochondria, and if you consume too much alcohol, you inhibit the mitochondria and a thing that's called the SER, which is the smooth endoplasmic reticulum, which is also another organelle inside the cell. So if you consume too much alcohol, you inhibit this mitochondria and SER, so you don't get oxidation of ketone bodies, and you get a buildup of triglycerides, which all convert to steatosis. Okay, we'll move on to now the next example. So for our next example, we have an intracellular accumulation of cholesterol and cholesterol esters. So foam cells. So a lot of cholesterol and cholesterol esters is we have these macrophages, these cells that phagocytize, or they gobble up, debris or bacteria, or they're just kind of the garbage man of the body. But these foam cells, what a foam cell is this cell. Let's say this tissue undergoes necrosis. So some part of this cell underwent necrosis, so all this tissue is dead. So these phagocytes will come in here and they'll start gobbling up all this. They'll start consuming and eating up all this dead stuff, so it's not just lying around our body. And foam cells are when these cells accumulate too much, or they eat up too much cholesterol, and so you start getting these vacuoles, these little bubbles, or these little bubbles of lipids, or sorry, of cholesterol. So that's what these foam cells are. Is they look like this under the microscope because they consumed a lot of necrotic tissue that has a lot of cholesterol in them. Atherosclerosis, I'm sure we've all heard of this. Let's say you have an artery here. Well, atherosclerosis is when you start getting all these cholesterol buildups inside the lining around the blood vessels. So then they start closing down on you, and they have kind of a yellowish appearance. I should color this in yellow. They start having this yellowish appearance, and then that is the process of atherosclerosis, and you also see kind of this narrowing here. Another pathology that you see with cholesterol is you have these genetic skin disorders, or sorry, these genetic disorders that are called hyperlipidemic syndromes, and they have too much lipids, but these macrophages will come in here and they'll be just saturated. They'll just be filled with, again, these little vacuoles, these little bubbles or pockets of lipids, and these xanthomas under the epithelium. So if you have a skin here, this is your skin here. These are the layers of your skin, and then below that you have extracellular matrix. You have you have collagen all over, but they'll start accumulating under here. These cells will start accumulating, and you'll start to see a little xanthoma from, if your eye is here and you're looking down at this skin, you'll start to see kind of a yellowish appearance, and that's called a xanthoma. Sometimes they can get quite big and they become bumpy, and they're just accumulation of these macrophages that have too much cholesterol in them. So there are several examples of protein accumulations. So another way that you can have intracellular accumulations is through proteins. Proteins can accumulate in cells. One example is, let's say we have a kidney here. And inside the kidney, you have the renal arteries coming in, which is blood. And you have some veins going out, which is blood. So the kidneys filter your blood. And inside, this is not anatomically correct, but just so you guys can have an intuition or a conceptual idea of what's happening inside the kidney, is you have this blood, you have the blood coming in that's, has wastes and byproducts of cellular respiration, of cellular function. So your body needs to get rid of that. So you can pee out the the bad stuff that needs to get out of your body. So you have a thing that's called a glomerulus. Okay, so the blood's coming in, and what it will do is it kind of passes by this glomerulus, and it kind of filters out. Stuff will get. It's like a big screen here with like small little holes in it, and stuff will come in. Will come into this glomerulus. Like if I had a screen here, let's just, if you've ever, gold miners, for example. Gold miners, that's searching for gold, they'll have screens, and they'll have certain types of screens that have certain holes, certain sizes of holes in them. And then they'll put dirt on here. They'll put dirt on here and they'll shake it, and only the stuff that's small enough to fit through these holes will shake out into a pile of something. And then they'll have another screen. They'll pass that through another screen, with even smaller holes. And then they'll shake that. They'll put this dirt on top of this screen, and then they'll shake it out, and then they'll get a pile of even smaller particles. Well, that's the whole idea of this glomerulus. Is the glomerulus has a certain hole size to it, if you will, and only particles that are small enough will filter through this. Like, you don't want red blood cells. One of the biggest proteins or cells, actually, you don't want that filtering through your glomerulus, because it will clog it up. And then it comes down. It's called the loop of Henle. You can review. I'm going to make a video in the physiology section about the glomerulus, but just so you kind of have an idea, is the blood will filter through this, and then it will get reabsorbed. Cells will reabsorb all the good stuff that you need. So the good stuff gets reabsorbed, like glucose and other things. They'll get reabsorbed, and it will go back into the blood coming out. So this is blood coming in, and this is blood going out. It will get reabsorbed back into the blood going out. And the stuff that you don't need, you excrete it out through your urine. So that's that's kind of just in a nutshell what the kidney does. And there's a pathology called nephrotic syndrome. And nephrotic syndrome is where there's something wrong with this screen here, or this glomerulus, so proteins that are bigger than normal are getting passed through this glomerulus. And so you have these cells being like, oh my goodness, we can't be leaking out all this protein. And there's a if you do a urinalysis, if you go into the doctor and they do a urinalysis and they say you have too much protein in your urine, there's something wrong here where there's not the only way, but one of the causes is this nephrotic syndrome where there's too much protein leaking out through this glomerulus, and that's called proteinuria. You have too much protein in your urine. Well, anyways, these cells are going to be like, hey, there's too much protein coming out of here, so we got to start absorbing all this back in. And when you absorb all this protein back in, these cells, these kidney cells, they start getting too much accumulation of proteins, and you can actually do a histology slide and see this under a microscope, and you can kind of see all these proteins inside these kidney cells from a nephrotic syndrome, or well, not from nephrotic syndrome. Usually you test nephrotic syndrome from a urinalysis to see how much protein you have in your urine. But anyways, these cells can start accumulating all this protein. Another example is, just a second. So another example of protein accumulations are Russell bodies. So what Russell bodies are is that, say you have a B cell. B cells are part of the immune system. Here's your DNA, and it will turn into a plasma cell. Oops, sorry. Space there. A plasma cell. It will turn into a plasma cell, usually in a lymph node or something like that. So what plasma cells do is that they're part of the immune system, and we'll talk about in future videos the immune system. We'll go through all the cells and what they do and all that, but for now, a plasma cell is a cell that produces antibodies. So what a plasma cell does is that when you have an endoplasmic reticulum, and then you have these ribosomes on top or adjacent to or connected to the endoplasmic reticulum, that's called a rough ER. Rough endoplasmic reticulum. Well, you start seeing all these immunoglobulins build up inside the rough ER, and that's usually inside of a plasma cell. That accumulation of antibodies or immunoglobulins is called Russell bodies. Another example, let me go down here. Another example is Mallory body or alcoholic hyaline. This is highly characteristic of alcoholic liver disease. So you have here in the liver, when people consume too much alcohol, you have Mallory bodies, and inside the liver you have all these different cells, and they think that it's inside the cell there's filaments that kind of help maintain the cell integrity or the cell structure. Inside these you have these filaments, and they think that it's an accumulation of these filaments, because when these filaments when the cell needs to adapt or move, these filaments are broken down. They're kind of let me get the eraser here. They're broken down and they're erased, but these filaments don't like to be degraded. And so you get these accumulation of these intermediate filaments, and that's what causes the appearance of a Mallory body, and that's due to alcoholic liver disease. So the next one is glycogen. Next one is glycogen intercellular accumulations. So in diabetes mellitus, you have problems with the metabolism of glucose. So inside the cell, you start seeing glucose glucose molecules being accumulated, and that's a problem with diabetes mellitus is the metabolism of glucose is some way dysfunctional. Also, there's a class of diseases called glycogen storage diseases, and um, let me just do this. So glycogen storage diseases are a class of disorders that have problems with storing glycogen, and we'll talk about some of the specific details of these glycogen storage diseases. And the next class is, and the last group is pigments. So pigments are, for example, melanin, which is in your skin when you're subjected to UV light from the sun or other sources. You have within your skin, you have these skin cells and melanocytes. These melanocytes produce the melanin, and they start producing when UV attacks them. They start, or passes through them or hits them. They start producing more melanin, and then that's how we get a tan. Another description is, remember when we talked about at the start of the video, we talked about how when you inhale carbon, for example, this is supposed to be your lung, your trachea, and your bronchi, but when you inhale carbon, you have these inside your lungs here, you have these little phagocytes, and they have little, this is a carbon. The phagocyte will ingest this carbon and bring it inside, and then you'll start getting accumulations of carbon. In coal miners, the disease is called pneumoconiosis. So that is a specific disease that is caused when coal workers have too much carbon in their lung. Also, when you inhale pollutants, or if you live in a huge city and you have a lot of smog and you inhale a lot of carbon, you can also get lung problems that will inhibit that will inhibit your ability to breathe, like emphysema. And so what happens is that these phagocytes, they ingest all this carbon, and then they're transferred to a lymph node in your neck or in your chest somewhere, and they can start collecting, and then you notice that you have a black or a dark patch on your skin, or that's just a accumulation of carbon because you've been exposed to coal or you've been exposed to urban life. And the last example is, we will get here blue, of pigment accumulation is a bruise. When someone hits you, or you get hit by something, or you hit something, you get a bruise in your arm, let's say. So I have here, and there starts to swelling. That's a lot of swelling, but nevertheless, you get a little patch or a bruise. And what happens is that the red blood cells, you got phagocytes inside here. This is blown up picture of here, but you have a phagocyte. And just in the case of this phagocyte consuming carbon in your lungs, these phagocytes will come in here to the area that's damaged, and they'll start consuming all of these red blood cells. And inside the red blood cells, you have hemoglobin. And inside hemoglobin, you have a heme, or an iron compound. And when these phagocytes start accumulating the iron from your blood, that starts turning, that's the color changes, and the color changes from like blue to green to yellowish, those are just changes within this process of iron metabolism and these phagocytes consuming these red blood cells and the iron accumulation inside the cell. So that's it for intracellular accumulations.